Good morning, everyone, and welcome to the first webinar of BioMeca. Today I will speak about atomic force microscopy, of course, but also fluorescence microscopy and how these techniques are able to offer you new perspective for your research. To begin, I will introduce myself. So I'm Gael Runel. I've been a bioimaging engineer at BioMeca since 2017. And I'm focused on the optical microscopy, but I use atomic force microscopy, of course. And I work to join these different techniques to go further in the sample analysis. And Céline Pallier is with me during this presentation, and I will let's introduce herself to you.
Oh, hello, everybody. For them who don't know me, I am Céline Payet, Business Development Director at BioMeca. I'm very glad to welcome you today at our first webinar. And I'm here as a moderator. That mainly means to check your questions on the chat and try to answer it in live or at the end of the webinar. So feel free to use this chat during Gael's presentation. Now I'll pass it over to Gael. Enjoy your webinar.
Thank you. So first, I would like to introduce briefly the BioMeca imaging service, the bioimaging, for of BioMeca. It is a support service that's very powerful to enable you to provide tomorrow's technology for your research. Our service is made up with different parts. The first one is to adapt a new protocol in a different type of microscopy, from atomic force microscopy to optical microscopy. The second part is data analysis from this different technique to go further in the outputs we can offer you. And the third part is the automatization of our process with a new device setup to adapt a sample or experiments and eventually manage data to go further in the data extraction and representation. So we will start the webinar by talking about atomic force microscopy and then multiphoton microscopy, and more especially about second harmonic generation, and finally with light sheet fluorescence microscopy. But before starting the AFM presentation, I would like to introduce different microscopy techniques with this slide in order to show you what kind of studies we can carry out. The first technique, and the more common, is the optical microscope. With this group, we find different kind of microscopes able to study from samples from millimeters as a tissue up to large protein, about 100 nanometers or less, with a super resolution microscope. Then there is electron microscope, which is very interesting for its high resolution power, but only for the small sample. And with this technique, we can study sample from micrometers up to the angstrom, but it is a very long preparation to study a biological sample. And we have the atomic force microscopy, which is a very versatile techniques because you can study very large sample, about millimeters, as this Medaka fish, up to very small sample, some nanometers, like DNA. And that's without any preparation and tissue alteration. So, okay, now we can start with AFM, and I will speak about the principle of the AFM, and then I will present you some studies on the skin explant, cryosection, and cells cultures. But the first question is, what is an AFM, an atomic force microscopy? And I'm going to introduce you two of our AFM devices. The first one here is a Bruker bioscope result. It is very fast for the acquisition and have a high resolution. Moreover, it is very flexible for the samples, and we use it for the skin explants, cryosection, hair, biopsies, and others. The second AFM is a Bruker multimode, which is the most powerful AFM in resolution, but able to study only small samples. And it's mainly used for liposome studies, proteins, corneocytes, and others. As you can see in this picture, we have different parts on the AFM. The first part is the optical microscope with a fluorescence system, and we use for the transparent sample and the fluorescence sample staining. Then there is acquisition plating with less than 1 nanometers precision to move the sample in XY axis. Here there is another camera with a LED for opaque sample, and we put the camera on the head of the AFM. And finally, the head of the AFM with the magnification here for the resolve and here for the multimode. And at the center of the head, we can find the holder where we put the cantilever. And as you can see on the holder of the multimode, the cantilever is here. And this cantilever is very important because it's the tip of the cantilever which is in contact with the sample to indent it. But what is an annotation? To explain it simply, we will use this movie with a golf ball here and the truck here. And when the truck drive on the ball, it applies a force, and this force deforms the ball, which becomes flat. And at the end, the ball comes back to its original shape without damages. An annotation is the same thing. We exert a force on the sample and to penetrate it, to deform it, and then the sample comes back to its original shape without any alterations. Now to come back to the AFM, I will show you this very simple picture. As you can see here, we have a cantilever with a spring constant registered, on which we can apply a no force that allows the move of the cantilever to indent the sample. To process the signal, we use a laser beam which hits the cantilever and reflects on the photodiode to 4 quarters. And we record the shift of the photodiode on the shift of the laser beam on the photodiode. And we obtain the raw data, as you can see here, with the force indentation curve. And there is different parts on this curve. The first part here very flat, which corresponds to the cantilever is not in contact with the sample. Then with this exponential curve, which is the indentation of the cantilever in the sample, and the red curve with at the end the little shift, which is the refract curve, and this shift corresponds to the adhesion of the tip of the cantilever on the sample. And then we've got precisely the contact points that represent the points where the tip touches the sample here. And from this position, we fit a theoretical model to convert this curve in a Young modulus, and the Young modulus representing the sample stiffness. And to calculate accurately the Young modulus, we use different theoretical models and different shape of tips. Indeed, as you can see here, we have different kind of tips with a sphere, conical sphere tips. Here with conical tips, and here very sharp tips. With a spring constant from 0.06 Newton meter for very soft sample to 200 Newton meters for very steep sample. And here, this kind of tips with conical sphere tips with large sphere diameter are widely used to study explant sample because the radius of deformation is large, and we can obtain a deeper Young modulus. The conical tips are very interesting for the cryo section because the mechanical resolution is way better than the spherical tips, of course, but more superficial, and that allows to record high resolution picture. And this kind of tips, very sharp, are able to obtain a very resolutive image as a transmembrane cytoplasmic pores, for example, or DNA. Then to perform the experiments, we have different kind of acquisition mode. The first one is a contact mode. And with this mode, the tip is all the time in contact with the sample. And the force applied remains constant because of a piezomotor, which controls the tip's height and adapts its position. And with this mode, we obtain only the topographical data of the sample. The second mode is a quantitative nanomechanical mode or tapping mode. And with this mode, it's very powerful because as you can see here, the tips oscillate on the sample and hit it punctually. As a contact mode, we define a threshold force, and the piezomotor adjusts the position of the tips to keep it constant. Consequently, we obtain in the first time the topography of the sample, and then the biomechanics, because each pixel of the picture is a force orientation curve. And finally, to process our force orientation curve, we can use different theoretical models. And I present you two models today here. The first one is a Snedden model, more used with conical tips. And the second one is a Hertz model, preferred for the spherical tips. Okay, now we know how an AFM works. I will present you one of our study on the aging effect. And for that, we will speak about global skin explant under controlled environment, then a live force tomography, then a skin cohesion, cell-cell cohesion, about mechanical and morphological dermoepidermal junction properties, and finally with a traction force assay. Okay, for this first study, we were interested about human skin explant. Indeed, for this project, we want to study the effect of the aging on the skin. For that, we have performed a matrix of force orientation curve here on our fresh in vitro culture under a controlled environment, because we had to keep this skin for another experiment. Here we processed our data on the average of the skin to measure the impact of the dermis. Therefore, we processed all the curve from the contact point to the end of the curve and fit the theoretical model. Thanks to this process, as you can see on this graph, the stiffness decreases with aging. Then, to go further, we process our first orientation curve with another technique called the live force tomography. And this technique allows to obtain a 3D mechanical representation of our sample. And to perform that, we process our force orientation curve with a segment with the same size, and that more and more deeper on the sample. And for example, on this slide, we have the contact point here, and we work with the first segment. We fit our theoretical model to obtain the Young modulus, and we go to the next segment. When we process all the curve with this technique, we obtain a 3D representation, like a stack picture from confocal, if you want, but which are representing the biomechanics of the sample. Consequently, we are able to obtain a great overview of the samples, and thanks to this 3D representation, as you can see here with the dermis, here the epidermis, and the dermoepidermal junction and the dots. Then, from the same skin explant, we carried out 20 micrometer cryosections to study skin barrier properties. And we perform very high resolutive images of force orientation mapping on a section, and we focus on three characteristics of the sample. One, the topography, then the stiffness mapping, and finally the adhesion force. On the topographical image shows the stratum corneum here and the epidermis here. And on the stratum corneum, there are different layers. With the second picture is the stiffness mapping. We can observe the same different layers here. And when we merge the topographical image and the stiffness, we can confirm that these multiple layers are the corneocytes. The last picture on the right here with the adhesion force mapping still show different layer, but between the corneocytes. And it is the intercorneocytic cement. And thanks to this analysis, we are able to study skin hydration. Because indeed, when the adhesion force increase is due to the lipid composition and the quantity of water in the intercorneocytic cement. And more we have lipid, more the skin barrier keep water. And we also can observe that the corneocyte stiffness increase with age, and this is due to the desquamation rates. Now, after studying skin barrier properties, we are interested in cell cohesion, and for that, we perform a resolutive first indentation curve on epidermic cell cryosection. To focalize on the greatest position, we use nucleus staining to perform our measurements. The first picture here represents the samples topography, with here the nucleus area of the first cell, here the nucleus on other cells, and here the desmosome between the cells. On the second picture, we have the elastic modulus here from young and old skin again. As you can see on this first graph, the tension between cells increases, but when we compare the cell junction, so the cell area between cells and the number of desmosomes between cells, we can see a decrease between this area and the number of strings. And we can conclude that when we age, we lose desmosomes and the tension applied increases on desmosomes. Now we will go deeper in the skin by studying dermoepidermic junction. On this picture, we merge the fluorescence microscopy with atomic force microscopy imaging. The DEG was labeled with collagen 7, and the biomechanical measurement was performed with a first indentation curve. Here, each pixel is a first indentation curve, with on the left the young skin and on the right the old skin. And as you can see here, we have a disorganization of the DEG because the young skin DEG is flat, whereas the old DEG is wavy. Moreover, we can observe the increase of the stiffness of the DEG with aging. And we compare the mechanical and morphological behavior of the DEG with a very close phenotype as aging. And finally, we studied the dermis with fibroblasts. The first picture is the fibroblast cells on a collagen matrix with topography mapping. Here, the fibroblasts are shown with a red arrow and fibroblast extension with a green arrow. And around, it's collagen fibers networks. Then on the right picture, we observe the mechanical property very close to the fibroblast extension. And as you can see, when the fibroblasts create a tension of the collagen matrix, the collagen stiffness increases when we compare this area, and again with this area and here. And with aging, the fibroblast stiffness decreases and the tension exerted on the collagen decreases too. And therefore, we can determine activation degree of the fibroblast comparing with control. Okay, and now I will present you another kind of microscopy, the second harmonic generation microscopy. And again, it's a principle and several studies we did with this technique. But before explaining second harmonic generation, we must understand the multiphoton microscopy. And the first question is, what is a multiphoton microscope? I think everyone know confocal microscopy, which is a very powerful technique to obtain a 3D images of sample using a fluorescent probe. And that is an example of photon excitation. The difference between confocal and multiphoton microscope is a type of laser beam, because a multiphoton microscope is done with titanium sapphire laser, which has a pulse width of 200 femtosecond, whereas a confocal microscope is done with a traditional laser, such as argon laser or diode, actually, which emits visible light. The second question is, what is the difference between these two laser beams? When we use a confocal microscope, when photon excites the electrons of the fluorophore by hitting it at the excitation wavelength, then the electron reaches excitated state, and when it comes back to its ground stage, there is a loss of energy released as a photon of light. In contrary, when we use a multiphoton microscopy, the first photon hits the probe electron at the half of the excitation wavelength. It reaches an intermediate state, and it's when this is a hit of the second photon which puts the probe in the excitated stage. And when the electrons come back to its ground stage, there is a loss of energy released as a photon of light. But what are the advantages of the multiphoton microscopy? The first advantage is a higher resolution of the focal plane illumination. The illumination area with a confocal microscope is a conical shape. It is larger and we need to use a pinhole to focus on. Whereas with a multiphoton microscope, the illumination area is smaller. We don't need to use a pinhole because the laser beam is very precise and there is less phototoxicity and photobleaching. The second advantage is the high wavelength of the laser beam. As it's twice higher, the beam penetrates deeper into the sample. Furthermore, as it's a lower frequency, it is much appropriate for the biological samples because it means less damage. And finally, of course, it is compatible with second harmonic generation acquisition. And now the question is, what is the second harmonic generation acquisition? So the SAJ requires an intense laser beams going through the materials with a non-centrosymmetric molecular structure. It is very efficient to observe a well-organized structure as collagen or myosin networks, for example. This signal comes from two excited photons which interact with material, and then a new photon with half of the wavelength is emitted. With this technique, a two-photon process, but unlike TPEF, SAJ is energy conserving, because which means it doesn't involve exciting molecules. For example, with this camera, the TISA laser generates a beam at, for example, 800 Newton nanometers. This beam crosses a telescope and its diameter increases. Then it hits the dichroic beam splitter, cross the lens, and interacts with non-centrosymmetric sample, which emits a light at 400 nanometers wavelength. And this is a half of the wavelength entering the sample, which means that the frequency double. And this is the second harmonic light. As we are in refraction configuration, light is emitted in the same lens. It crosses the beams, the dichroic beam splitter, and hits the photomultiplicator to acquire the signal. Okay, let's move to the use of second harmonic generation. We will go through different studies to demonstrate how it is useful. For that, here we studied the collagen density and the number of dermal papilla in the skin and in the skin explants. We put the fresh sample into the microscope under controlled environment, and then the sample is penetrated up to 54 micrometers, and the stack image are acquired. This slide show acquisition for a young skin in the first row, and for the old skin in the second row. The collagen fiber are showed in science. The yellow color correspond to the tissue autofluorescence. And after several setting and picture analysis, we conclude the conclusion is clear. The collagen fibers density and the number of dermal papilla decrease with aging. Now, to confirm our results, we study the difference of collagen density between young and old skin using cryosection and atomic force microscopy. So the collagen fiber are shown in cyan, The the yellow color is elastin, and in green it's epidermis cells. As you can see on these charts, there is a decrease in the collagen density with aging. But more interestingly, this decrease is highly correlated with skin stiffness obtained with atomic force microscopy. And to better understanding of this mechanical results, we studied the organization of the collagen fibers in the dermis. And we performed atomic force microscopy, quantitative nanomechanical imaging to analyze the fiber orientation the first time, and then the collagen period and the collagen stiffness. And we compare young and old dermis here. As you can see here, the collagen fibers in the young dermis is more anisotropic than the old dermis. And also there is a slight decrease of the collagen period and the collagen thickness with aging. Now on this slide show our study on the production of the collagen by fibroblasts cultured on petri dish between a young and old fibroblasts. And to observe the production of collagen, we put the collagen on petri dish with a fibroblast for 4 days. Then the fibroblasts were fixed and stained with phyloidin, which tags the actin cytoskeleton. And then the sample were analyzed with a multiphoton microscope. Here we merge the multiphoton image with a second harmonic generation image in order to match the fibroblast cytoskeleton and the collagen production. To see underneath the fibroblasts, we virtually remove the fibroblasts and keep its imprints. And we observe that the collagen density is higher underneath the fibroblasts. Then we extract the collagen ratio with quantity underneath the fibroblast and around the fibroblasts, and we compare it between young and old fibroblasts. And the result is shown on the right chart here, and we can observe the decrease of the collagen production with aging. And then we observe the fibroblast shape, you can see here, with the aspect ratio. And as expected, there is a decrease of this parameter for the old fibroblasts. On this slide show our new solution to study the morphometrical property of tissue. And as shown earlier, the laser beam of a multiphoton microscope goes deeper in the sample. And here we acquire a stacked image of a skin explant with cortical actin staining on this picture. Then we processed this stack image to create a 3D numerical reconstruction with this picture of skin with a basal layer here and the more external layer and these parts. Then we carried out 100 micrometer 6 cryosection and stained the cell and nucleus to obtain a 3D numerical reconstruction again. And finally, we perform the same process with a fibroblast on a dish. And now what is interesting here, with this technique enable us to observe the cell organization, the cells and tissue volume, and the cell-cell contact surface, and other properties. And finally, my favorite picture here, because we perform AFM mapping and high-resolution imaging with only one section. With AFM mapping enable us to observe the mechanical property between the derm and the epidermis, while the high-resolution imaging enable us to observe the collagen fiber orientation, and here the collagen thickness and period. Then we quantify the collagen and elastin dermis density with TPEF and second harmonic generation. And finally, we develop an algorithm which enable us to segment the epidermis cells and extract cells metrics such as volume, morphological shape, and cell-cells contact surface, for example, and others. Okay, now let's go on with another optical microscopy techniques called the light sheet fluorescence microscopy, and we will go through its principle and some uses. So with this microscope, we must forget the basic schematic of the microscope, because no ocular, we cannot see the sample, and the sample, which is a sample which move to make the acquisition. Like a confocal microscope, the beam is based on the fluorescent probe and is able to acquire stacked image. But how does it work? So we have a laser beam which closes a special lens which produce a very flat illumination, and that's why we call the light sheet microscope. And then this illumination sheet lights up the entire focal plane of the sample, and the picture is acquired with a second lens located at 90 degrees angle and with one CCD camera. And this technique is very interesting because in addition to move the sample along the XY axis, we can rotate the sample. So we can do acquisition at 360 degrees. But firstly, we need to prepare the sample to make it transparent. This is a technique requirement because the sample is very large and we need to have a transparent samples. And then we embed the sample in agar, for example, with this Medega fish here. And I show here the prepared sample is put in the medium chamber of the microscope. So the SPIM here is a little bit different from the previous schematic because we have two illumination paths which oscillate to reduce the shadow effects when during the acquisition. And then we acquire image from different acquisition angle, and then the images were processed to obtain a very resolutive picture. So for this study was done on the whole body of the Medega fish, here on the left picture. And here we can observe the muscle fiber on the top. And here is the anal fin of the Medega fish. I'm sorry. So on this picture, it's a magnification of this area. And as you can see, we can identify the muscle sarcomere here, and here the epidermis cells. Okay. And this technique is ideal to study tissue organization, cell morphology, and morphometry properties, and dermis and epidermis junction properties, and finally, whole tissue or cell stain organization. And finally, the conclusion with this tab. And I show you an overview of techniques approach on this webinar and the other study carried out on the samples like skin explants, cross-section, and cells. And of course, we haven't yet developed all the potential of the techniques at this time, but that will be the subjects of the next BioMeca's webinar. And I hope this webinar has been interesting for you, and I thank you for your attention.
Okay, you're done. So thank you all for your attention. Maybe we can check if I have some questions. Let me show. I had a private question about. I understand that you work on fixed cells with ACG, and could you work on free cells to follow a matrix production with this technology? So if you can answer, Gael.
For the kinetics of matrix production?
Yeah.
Yes. Okay. Yes. Of course, we can work on a fixed cells with second harmonic generation, and if we want to work with primary cells, we have to stain cells But if we use vital DAPI or other tracker staining, we are able to follow the kinetic of the matrix production. And for a cell line is easier, of course, because we can use life facts, for example, or others. And moreover, we can fix cells at different time points and compare the collagen production. So yes, we can do that.
Okay. Maybe last question was about how could you? Sorry, I will choose another one because it will help me to introduce something. How could you study skin microbiome with your technique?
I'm sorry. Can you
How
repeat,
could
please?
you study skin microbiome with your technique?
Oh, okay. Um, okay. I think the next webinar will is very interesting, of course, because the AFM spectrometry is very powerful for that. And with this technique, we can stick antibodies or chemical probe on the tip, and we scan the sample to characterize it. And yes, it's a very powerful technique, and we can do that with AFM spectroscopy and the functionalization of the tips.
Yeah. Okay. So thank you very much, Gael. It was easy for me to conclude this webinar because I'm delighted to tell you that we plan another one at the summer's beginning, and the subject will be atomic force spectroscopy, innovations and opportunities in life science. So we wish to welcome you again. And be sure we'll let you know these new webinar details as soon as we set a date. So I like to thank you again for your attention, and I wish you a very good day, and of course, be safe. Thank you all. Bye.
Thank you. Bye.